To start with, I'm going to try and summarize the idea behind this talk for you. Hopefully you guessed from the title, it's about trying to give you the sort of a 10,000-foot view of analyzing DOE data. Specifically, I want to do three things. I want to talk about how design and analysis are related. I want to give you a mental model for the process of analysis. And I want to give you a bit of information about some of the most important techniques you'd normally use, along with signposts for where to go to find out more. So to start with, I'm going to give you a bit of background on how designs work. Some of this could be familiar to some of you, particularly if you watched any of our earlier webinars, but it's important context for understanding analysis. So as you can see, I've chosen to use this bust of the god Janus here to depict this idea. And what I'm really trying to say here is that essentially designs and analyses are two faces of the same thing. So to start with, let's talk about the sorts of experiments that you can do. Essentially, I tend to think of them as coming into two sort of broad types. You can be trying to learn something about a system of interest, or you can be trying to find a way to get it to behave in a particular way. And there's two broad disciplines which cover these activities, modeling and optimization. Modeling allows us to represent our specific idea of how things work inside our system, and optimization allows us to make use of that model to identify how to get it into a particular state, if that's what we're trying to do. And DOE is largely about trying to use modeling techniques to get enough of an understanding of our experimental system to work out how to get it to behave in the right way. So I've talked about this idea of an experimental system in the abstract, but I think it's really useful to frame the discussion in terms of a more specific example without losing generality. So I'm going to quickly run through one. So this is a classic analog synthesizer, the ARP 2600. Probably doesn't look too familiar to most of you, but that's fine. It doesn't need to. Let's just say this is what we're interested in investigating. This is our system. My goal in introducing this to you isn't to teach you about subtractive synthesis, but to help explain why DOE exists and introduce a bit of terminology for those who aren't familiar. Understanding this motivation is really key to understanding how designs and analyses work, because you know what sort of problems they're trying to solve. So this thing's a big black box with lots of controls on it. 140 so buttons, knobs and sliders, which all have an influence on the sound the synthesizer generates. In DOE terms, these would be our factors. If we're looking to understand how the controls work or just produce a particular target sound, we can set the control somehow and press one of these keys and observe what happens. In DOE terms, this would be a run. We can then listen to the sound it generates through the speakers. From this sound, we could identify certain properties such as the loudness, the overall timbre, and use these to generate some responses to say how close we are, which we can use to say how close we are to our target. A key point about this system is that setting these controls isn't optional. They all have to have some value. And this is a bit overwhelming if you're experimenting. It's obviously, there's a huge potential space of possibilities to explore. If we follow the conventional idea of changing one thing at a time, we hit the issue that whichever controls we're not changing still have to be set at some value. And this causes a couple of problems. Firstly, some controls actually depend on others for their behavior. In this case, I've highlighted the filter, which has two main controls, a cutoff and a resonance. At high and low values of cutoff, the resonance doesn't really have a perceptible effect, but at intermediate values, it makes a very significant difference to the sound. In DOE terms, this is what we call an interaction. Note this isn't actually one of the factors, it's a more abstract thing. It's a way that two factors combine to change the response, which in DOE terms would be called an effect. When we come to talk about building models later on, we'll usually be talking in terms of effects rather than factors directly. In case you're wondering, the effect of a factor on its own is actually called a main effect. Another issue we can hit is that there can exist controls which completely kill the response, regardless of anything else. For example, I've highlighted here a switch that turns the speakers off. If we explore with this in the wrong setting, we won't make any progress at all. Okay, so fine, what's the relevance of all this? Well, I hope you can recognize in this that there's a lot of characteristics this box shares with some real interesting systems. They usually have the same sorts of characteristics. They're subject to lots of external influences, which can have a variety of different effects, often with dependencies between them. And very often, there are some you can completely kill behavior with if you set them wrongly. Whenever you work in a real system, you're setting all of these influences to some values in most cases, whether you mean to or not. You have to experiment at some temperature in your lab, regardless of whether or not you explicitly set the thermostat to some value. Experimenting in parallel on as many as you can is a key way to de-risk your experiment. A second thing that DOE does to de-risk experimentation in complex systems is using iterative methods. The typical stages used are depicted here and follow a course started with screening stages through to optimization. In screening, the goal is to try and understand which factors have the biggest impact in your system. And at this stage, we're changing as many as at once as possible. So for example, we might start with say 15 as a reasonable number. Later on, we then move through to focusing down on a very small number of extremely important factors, 4 in this example, which allows us to then do an optimization and actually find a good result. By following an iterative process like this, we can handle more factors and also are more insulated against cases where we make bad choices, in which case we can try again. What's important here for analysis is that the goal of what your analysis is trying to achieve at each step varies depending on where you are in this process. At earlier stages, you're mostly trying to define what experiment to do next, which is very different to later stages where you care about actually making a good model and getting good predictions. So typically another feature of our systems that you'll often find there's some sort of prior data on it, which is usually arisen through some kind of selection process. For example, with our ARP synthesizer, there's published control settings or patches for achieving particular sounds. These sorts of things are usually readily available and might be another way to get an insight into our system, but there's an important distinction between observational and experimental data that must be borne in mind, which is that there's usually a lot of bias in the kind of things that you observe out there in the wild. And it's really impossible to understand cause and effect effectively. One usually cited example of this is the sort of correlation is not causation kind of idea is this graph, which purports to show that pirates keep global average temperatures low. The key problem, of course, is that both are changing over time, alongside lots of other changes which are actually affecting global temperatures. The great thing about experiments is that they prevent you being in situations like this because you're freely changing all of the variables in question and avoiding biases. It's useful to know that most textbook examples of linear modeling will assume you're dealing with observational data, which introduces issues which you often don't have to worry about so much when you're analyzing DOEs. So that's something to bear in mind when you're reading up on this kind of thing. So I hope you now understand how the problems DOE tries to solve arise. So next, to really get to what's going on in analysis, it's important to understand how designs work and how their features affect the analysis. In general, for any design, there's two things we're trying to do. There's two main objectives. Firstly, we're trying to uncover the structure of cause and effect in our system. And secondly, we're trying to distinguish real signals from random variation. So let's return to the idea of changing one thing at a time. Why are we always told to do this? Well, the point of that is that really we can always say which effect must be caused by that change. It's obvious because you've only changed one thing. If you want to change many things, you now have a problem. How do you keep this sort of property of identifying which causes and causing which effects? The solution that designs use is to keep factor changes orthogonal, which means keeping things at right angles. And you can see this represented in this image in the top right here. In effect, what we're saying is that when we change two things independently, we sample all possible combinations of their levels. Probably the most intuitive way to think about this is in terms of the correlations between the two factors across all the runs in the design. So if we look at the lower image, we can see two factors here, temperatures at different stages of a process. And you can imagine calculating a correlation coefficient for these two columns. Two orthogonal factors will have a correlation of 0, whereas if they're completely dependent, they would have a correlation of 1. So, for example, if we had just two factors, x1 and x2, there's 4 combinations to investigate if we're only looking at 2 levels, which is common for the early stages. You can see them plotted here on the right. At the top left, you can see the actual 4 run values that have been chosen. And the top plot on the bottom left is particularly interesting. This shows the correlations between the two factors and each other, and also with their interaction, this x1 by x2 term. So you can see that x1, x2, and their interaction all have correlation 0, 0 being white in this picture, whereas obviously they're all correlated perfectly with themselves in black here. This is great. It means we can always distinguish how the factors cause variations in the results. However, unfortunately, the number of runs you need to do to perfectly resolve all the effects and pairwise interactions grows very, very quickly as you add more factors. So typically, early stage designs can't be as orthogonal as this, and they have to introduce compromises to orthogonality in order to keep run numbers reasonable. Correlation plots like this simple one down here are useful in showing us where these compromises are being made. So with 2 factors, there isn't much scope to see how this works out, but if we move to 3, we can get the idea a bit more. Here's the same plot for a 3-factor design with 8 runs. 8, being 2 to the 3, is the so-called full factorial design. So with this many runs, we can essentially see a clear picture. However, if we're willing to tolerate a bit less orthogonality, we can halve the number of runs and only have to do 4. Designs where we make this trade-off are called fractional factorials. 4 is half of 8, so this would be a half fraction. And here's what that correlation plot looks like. Notice we've got this cross pattern now. This means now that all the two-factor interactions are perfectly correlated with the main effects. This trick is known as aliasing. So in this case, we'd say the main effects are all aliased with the two-factor interactions. This is a common trick used to make DOE experiments efficient at early stages. When we're trying to get a feel for our system, we're going to bet that at least one of these more complex effects isn't actually active, equivalent to saying the two factors behave independently. The trade-off is that we can get into a situation where we've detected an effect, but we don't know whether it's due to, say, x1 or the x2 by x3 interaction. There's a few ways to deal with this situation. We can potentially arrange things so that those ties are easy to resolve because we have some a priori knowledge we can bring to bear on the matter. Interactions also tend to get less common as you include more terms, so there's usually a bias towards lower order effects, which is exactly why this design has this pattern that it does. You won't normally try to have correlations between main effects, for example. Of course, the only real way to know which is to do another experiment, and one conclusion you might reach in analysis is that actually resolving a tied situation is exactly your next objective. Factorial designs and their fractions only exist for certain choices of run number. However, we can apply a different design method, optimal design, and have exactly the number we want. This correlation plot is for a 6-run design for 3 factors. Notice that now, alongside the cross pattern, we have these boxes which in grey, which show some factors, x1 and x3, for example, are now partly correlated at 0.333. This is known as partial aliasing. Although it's obviously similar to full aliasing, there's an interesting difference. We don't really have to do anything to directly resolve these differences. What actually happens is we lose some ability to distinguish each effect from background noise. That's very useful because we're swapping some degree of sensitivity to effects without the potential confusion of trying to figure out which of a set of alias things is really active. One thing which hopefully occurred to you is that this plot's exactly the same as the last one, except now we have some extra correlations. If you look at it this way, this design's obviously worse than the last one, but we added 2 runs. So how did we come out with what's apparently such a worse result? And that's a great question. The answer is that if we look at things this way, it is indeed worse, but there's actually another dimension to things that we're ignoring, which is the question of power. So what's power? Power is the concept statisticians use to assess how well an experimental design can distinguish a specific effect from random variation. Imagine we're going to do a hypothesis test. On the left in blue is how the data would be distributed by chance alone. On the right in red is how the data would be distributed given the effect of a particular size, represented as the distance between the mean of the 2 distributions. Given a particular choice of critical value for the test and assumptions about the size S of the effect and the amount of variation epsilon, we can then calculate the chance that we could correctly detect a real effect, and this is what's known as power. In real designs, power gets very complicated. Design packages give you calculators to help you to understand what's going on. But typically, it's highest for main effects and successively lower as interactions get higher in order. And it's always a function of the design as well as the terms that you actually ultimately are going to put into the model. So to finish up on our question from before, here's what the power analysis looks like for the 2 designs in 3 factors with either 4 or 6 runs. First, look at the left panel. Hopefully you can see the various features we just talked about. There's a significance level for our t-test. There's an anticipated error or RMSE. And then for each term in the model, we can say how big we expect the effect to be. This is the anticipated coefficient. On the right, we get told the probability that we would be successful in detecting a real effect given these parameters. A typical rule of thumb threshold is about 0.8, but typically it's mostly just used to compare different designs rather than as an absolute measure. And so you can see now that with 4 runs, despite the lower degree of aliasing, we've actually got less than half the power in this circumstance. And just two extra runs has actually given us almost more than twice as much. So you can see actually the extra 2 runs really are doing us a lot of good here. Okay. So now we know a bit about what we're trying to do with designs and a little bit about how they work. We can use this knowledge to do our analysis. So the specific takeaways from the last section that I want you to remember are that typically we have several iterations in our DOE, and our goal when analyzing each stage will be different. Ideal designs have all factors and interactions of interest orthogonal, which means it's easy to distinguish cause and effect. But real designs nearly always compromise this, and this is important to know for analysis. Either we'll end up unable to distinguish certain combinations at all, or we may end up less able to distinguish some effects from chance. And typically this will be a compromise around the sort of model that you can ultimately fit. Now, I'm going to go on to talk more directly about how to do analysis, both in general, in terms of what I call analysis loop, and specifically in terms of tools like linear modeling. So first off, I need to tell you about an important general concept for all modeling. When you're building a model from data, you're typically trying to get the best fit, and you can make various different moves, such as including or excluding particular bits of data, trying different techniques, including or excluding particular effects from the model, and so on. To see how well any model is doing, you might use a measure like R² to assess goodness of fit, but it's important to be very careful here. The thing you need to know is it's nearly always possible to make your model arbitrarily good by just adding more and more terms to it. DOE experiments are particularly prone to this, since there's usually hundreds of possible effects you could put in. It's quite common to find things that look significant but are very small in magnitude. Things like this can be misleading, and it's important to be very careful. The best way to sum it up is to think of yourself as trying to walk a tightrope, balancing between including too many terms and including a lot of random noise, and including too few and missing the overall trend. You can see this depicted in the diagrams here. In the middle, we've got a reasonably good fit. There's some variation, but the overall shape is basically the same as the data, so it's about right. On the left, we've underfitted. We've fitted a straight line when there's clearly a curve in our data. On the right, we've matched the trend in the points a bit too closely, and I really wouldn't think this would generalize well. There are a number of methods you can use to account for this trade-off. There's some other measures like AIC and BIC, for example, but whatever you choose, it's really important to bear this whole concept in mind. So just here's a quick example from a real DOE. If you just run an automated model generation process and just let it run until it finds the best R². So yeah, this looks pretty good in terms of being a fit to the data, but there's 43 terms. I'd be extremely skeptical that any experiment would produce, biological experiment would likely produce data this good. Okay, so how are we actually going to go about doing our analysis? Well, at any given time, there's usually a few problems to solve. For example, you may have some terms that you want to include, which are obviously big enough to be included, but you'll often have some which are a bit more borderline. Alternatively, you might find your data doesn't quite match your assumptions or the assumptions required by your modeling technique. And this is often about removing some aberrant points. So typically in analysis, you tend to follow this kind of loop where you change the inputs to the model and see what conclusions you can reach. You might hypothesize that a particular pair of factors interact, for example, and try adding that term to see what happens. You can then do this a number of times, looking at whether your interpretation changes significantly when you alter some of the inputs. Conclusions which remain the same despite all these changes are much more likely to be correct. So the first step is to choose exactly which data to build the model from. This can include removing rows, perhaps to avoid outliers or zeros, which can dominate analysis completely, and potentially transforming factors or responses. This is usually a technical measure aimed at fulfilling model assumptions, and I'll go into it a little bit more later. When you first encounter the data, it's usually a good idea to get some kind of qualitative feel for what's going on. So the main idea is to get a feel for how the data are distributed. For example, you might want to see if there's more than one group. This can give you an idea if the modeling is likely to work well or if the data need a bit of attention before trying to model it. For example, some data sets have a lot of small values and very few extremely large ones, and these generally don't work very well for most modeling techniques until you do a bit of work to make the values more continuous. So here we have some data from an old experiment of ours from quite a few years ago, where we were investigating growth conditions in some engineered E. coli. There's 16 factors in the design, and we're just looking at the amount that the bacteria have grown. So on the top, you can see a distribution that I've created to show how our 64 different runs that we've done are distributed. And on the bottom, you can see how we would expect it to be distributed given a normal distribution of the same parameters. We can more or less say that these look all right. It's not exactly normal, but to my eye, it's not so bad that it needs any obvious attention. Qualitative analyses are also very useful in the event that your model building actually doesn't go so well eventually. This can happen when you first experiment on a new system and you get the levels wrong, for example. So here I've plotted for this same experiment I've just discussed, which is an example we'll be using right the way through to the end. I've looked at just what the top few best growing runs looked like, and I've tried to see if there's any particular pattern in how the particular input factors are distributed. Obviously there would be 16 of these if I showed you the whole thing, but it's quite busy, so I've just pulled out a couple to show you what it looks like when there's not much signal. So for example, in strain, there's nothing really to say either strain is doing much better than the other. And for post-induction temperature, there's some indication of a trend, but clearly for glycerol and the amount of time after we wait to harvest our E. coli, all of the top 5 are at the higher end of the scale of the 2 runs that we've tried. So this is really interesting. Since giving the bugs a bit more food and letting them grow for longer before harvesting seems like a pretty intuitive way to get more of them, it sort of makes me believe there is something in these kind of data. But of course, to really understand if something's having an effect, you'd also need to look at the lower end too. Do shorter harvesting times and lower carbon levels mean lower growth? So I've done a few sort of violin plots here that can show you roughly how the data is distributed for the 2 levels of the factors, and they can give you another way to see what's going on. Looking at this picture, it's not really quite so clear that whether or not there is much of a trend for glycerol and harvesting time. It actually seems possible to you now that this little group at the top that we're seeing might very well be the result of an interaction between these 2 factors. So having selected this data set, we then go on to build our model. There's a lot of possible techniques you can use, but for DOE most of the time, it tends to be a variant of linear modeling. The main choice here is which terms, main effects, interactions, quadratics, etc. you want to include. The choice can then sometimes be made automatically, but it's usual to make some kind of manual adjustments. Some modeling methods will also have other parameters which determine characteristics of the model or how fitting is done. In early iterations of a DOE, we usually have a large set of possible models which could be fitted, and we need some way to decide which ones look real based on the amount of random variation. So this plot's an example of a half normal plot, which is a very common tool for visually deciding which effects to include in the model. Essentially, orders are ordered in increasing size of effect from left to right. By chance, the factors would all lay along this blue line here. Bigger deviations from line suggest that there's more and more signal compared to the amount of background noise. And this is exactly where the power we saw earlier starts to play a role. We might perhaps be a bit surprised that some effects we expected aren't showing up, and it's possible that they may be a bit underpowered. So that's something you might choose to go back and investigate. For a slightly more objective procedure at selecting factors for the model, it's also possible to see p-values of significance. The process here is essentially doing one of sequential t-tests on the various terms. Looking at our growth example we saw before, it seems our initial guess was right. The most significant effect coming out is indeed an interaction between harvesting times and glycerol. And I can promise you this isn't something I looked at first. This actually surprised me. One important feature to notice is that there's actually 2 p-values in this table reported. And this is really worth knowing about. The left column, it's the individual p-value. It's just a normal set of t-tests conducted independently of each other. The column on the right accounts for the fact that by doing more and more t-tests at a given significance level, we're going to see more come out as passing purely by chance. So for example, if you specified a 5% significance threshold, 5 out of every 100 tests you do will just randomly test positive. So this corrected column is really, really useful in this situation where effectively what we are doing is doing a large number of parallel tests. It can be a bit conservative, but personally it's my go-to. And I would tend to be a bit more careful if with terms that only appear as significant on an individual basis, which starts to explain why I've sort of cut off the selection here. Having chosen the effect, we then have to run a model building process. For linear modeling, this is pretty straightforward. The most important thing to know is that there's a hierarchical principle which you need to follow, which to try and explain this, what it means is that if you include an interaction, you have to include all its sub-effects, even if they don't appear to make an individual difference. In this case, for example, if I want to include this harvesting time by glycerol interaction, which I obviously do, I would need to include both harvesting time and glycerol in my model. And this is something I have to do, despite the fact that based on its simultaneous p-value, I wouldn't ordinarily include glycerol in the model. I haven't got an easy way to show it, but I should mention that there are a few factors aliased with this combination to the interaction that I've selected. But I'm not investigating those for the moment because none of their subterms are coming out as particularly significant, whereas although it's a bit borderline, both of these are fairly high up the list. Here's what the model building window now looks like in JUMP. So you have to choose which terms to include and which responses you're predicting. There's a bunch of other options, but it's basically just a case of those things. One key decision you have to make at this point is which of a set of aliased effects you believe are active. Here at the bottom, you can see the eventual equation that we're trying to predict. What this says is that for each response J, we're going to fit a model with a constant term beta 0 and a set of terms which depend on the variables in the data, beta 1 through to beta p. Then finally, we have this epsilon error term, which is a random variable sampled from a normal distribution with mean 0. It's useful to see this equation and kind of bear it in mind because the betas are usually what you're actually interested in, since they're really what tell you about the structure of cause and effect in your system. So, having run our fit with chosen terms, the resulting model can then be inspected to see how well it explains the data and whether its various assumptions are satisfied. Typically, the parameters of the model, which usually give you an estimate of the size and direction of an effect on a response, should be examined and recorded for comparison with results of other analyses or different experiments. It's often useful to make predictions with the model at this stage as another basis for interpretation and comparison. The first question is usually how well the model fits the data. This can be assessed by plotting predicted versus actual values in XY plots and comparing the trend to the XY line. It's usual to consider some sort of goodness of fit measure, such as R-squared, but of course, as I've described before, it's really not sufficient just to do this. On the left, you can see a standard cautionary tale from statistics. This is the well-known Anscombe's quartet. All four of these plots have exactly the same R-squared of 0.67 for the line fits there, but clearly in 3 of the 4 cases, there really isn't actually a linear trend. There is only a good linear trend in the case of the top left. One of the most important assumptions in linear modeling is that you'll see a trend like this once you've made some predictions. And so this sort of check is really critical and often tells you most of what you need to know. For linear models, another key assumption is that the random errors of the data are constant. If you remember from the equation, this makes sense because there's only really one number that can describe how much error you have for every observation. Although it's not absolutely critical, it is worth verifying that errors are indeed constant or nearly so, otherwise you can easily miss effects. The best tool for doing this is a residual plot, some of which are depicted here. The plots on the left show how this looks when the errors get either larger or smaller as the response increases. Note there's a sort of characteristic cone shape, which is a telltale sign that you should do some sort of transformation on your data to try and bring them down more like this picture in the bottom, where you've got something approximating constant variance. Looking at the model in the upper right for our example, we can see it's not really doing too badly. There's clearly a bit of kinkiness going on in the middle. But as per the original interpretation, it's probably not going to be the worst thing for our results. So I wouldn't say this strongly indicates a need for any kind of transformation. A second thing that's really important is to look for outliers. These are points which substantially buck the trend and are very poorly predicted. And these can be a real problem because they can strongly affect the actual overall shape of the model. You can often spot them by eye, but there's also a version of the residual plot, which I've shown in the lower right, which can help you. Here we've got green lines that identify points which are significantly outside the confidence limits of our prediction according to the individual test. The red lines show the same, but for a test which is corrected for multiple testing. So there's a few points which are perhaps borderline, just over this green line here, but there's nothing really too bad. So although we could investigate these points, it's not really likely we'd see huge differences. The final thing we can look at with our model is then what it's actually telling us about the system. And to do this, we need to look at the parameter estimates. The column to look at is this middle one here, where we see estimate. This tells you the size and direction, positive or negative, of an effect. So we can see that from this that on average, both the effects of harvesting time and glycerol actually came out negative, meaning more is bad, but then we have this weak positive interaction term, which is here. Which is here. It's 0.1454. This means that we're seeing an unexpectedly good result based on looking at either of the two individually. So although they appear to reduce the amount of bacteria we get, if you set them both to high, suddenly we get a big jump up. Another thing you could do to interpret the model is to make a prediction. JUMP provides this profiling tool, which is a really good way to let you play with the factor levels and see how the predictions change. This model actually predicts that we should use low glycerol and a short harvesting time, which is really interesting and quite unexpected given what we initially saw when we eyeballed the data. So we now have the choice of whether to go back round, perhaps varying the data or building a different model, aiming to see if we draw similar conclusions. Typically, we'd expect to do this a few times, depending on just how bad the data are. Once we're happy with the model, we can use it by deciding either how to do the next experiment, make specific predictions to test, or stop and write up conclusions. So to finish off the example we've been following, what could we do next? Well, if we wanted to dig deeper into this data set, we could try transforming the response to make it more normal since it wasn't perfect. We could remove some of these outliers and redo the process to see if anything changes. Or we could investigate some of the other aliasing possibilities for the interactions we found. However, based on all our prior analysis, my overall feeling was that we wouldn't really get a great deal out of doing all of these things. None of these features really suggest anything deeply problematic. And since this is an early stage experiment, we're much likely to do better getting more data than torturing this data set anymore. So my choice here would be to stop modeling here and do another experiment. But what experiment should we do? Well, it's clear from the model that we could investigate using more NADPH, peptone tryptone ratio, and M9 salt time, along with using M9 LB as a medium. However, the choice for harvesting time and glycerol is less clear. The model says both low, but all of our best growers were high on both counts. So to finally decide, I made this plot, which splits the results into 4 quadrants with each of the 4 combinations. So you can see we have high glycerol on the right here and high harvesting time at the top. So if you have a short harvesting time, you can't add a load of glycerol. Similarly, if you have a long harvesting time, you must have lots of glycerol. You can do okay if you keep both low, but you can do a little bit better if both are high. This is a sort of shape that our model really isn't actually very able to fit well. Ideally, we really need a bit more data to help us get this relationship. So what we're probably seeing is down to 2 things. We've set the factor levels quite far apart, and we probably need at least a few other points in between the 2 extremes that we're sampling to capture more of what's going on. So my conclusion would be that we've got into a situation where our design doesn't really let us fit the right model. We can use this information in the next design, ensuring that we don't push the relationship so far and risk going over a cliff as we have here. Based on our results, I would then choose high for these variables. Looking again at our top predictions, they're largely consistent with the other model predictions, so it seems a good bet that there are good conditions in that region. So, to answer the question I started with, what really matters? First of all, understand your design and understand how it constrains your analysis. The sort of models you can build depend entirely on the limitations of your design. Your analysis might well be telling you that your design can't give you the right model. Second, keep the whole system in mind and bring all of your knowledge to the table. DOE is about de-risking exploration. The more you know, the better the questions you can ask. You can avoid really bad results like switching off the speakers in your synthesizer if you bring a little bit of prior knowledge in. Be wary of models that are too good, but also learn to spot when a model just isn't good enough. Be especially wary of automated model building, as it's often a quick route to overfitting. Modeling is a scientific exercise. You're following a loop of generating and testing hypotheses, essentially. Doing this a few times and looking for consistency between different analyses and keeping careful notes on what you've tried and why is really critical, particularly with very tricky data sets. Most importantly of all, keep the goal in mind. Some data sets will raise lots of questions, but you may not be able to answer some of them without gathering more data. Even when you have a rich data set, you often only need to know enough to decide which experiments to do next. You don't need to investigate every little detail. To quote Ronald Coates, if you torture your data enough, it will eventually confess to anything. And that's particularly true for DOE. My final thought to you is that while I hope this introduction has been helpful, it's been a very brief summary of a huge topic, and most importantly, one which can't really be learned just by hearing about it. It's pretty easy to get great tools and interesting data sets to play with. So hopefully if you get a copy of these slides, you can follow some of these links and try out some of these different data sets with whichever your favorite choice of software are. So finally, I'd just like to thank a few people who helped with this talk, Jar Jar, Marcus and Jane with discussions of titles and content, and most especially Amy, who did an amazing job of making my slides consistent and presentable. Thanks to them and all at Synthace for all their feedback.